Carolina Makowski. She is currently a postdoctoral fellow at UCSD in the Department of Radiology, and she has just completed her first year of a K-99 ROO NIH-funded application entitled Towards an Etiological Model of Adolescent Eating Disorders Through Neuroimaging, Genetics, and Behavior. She's also funded through a Young Investigator Award through the BBRF, looking at contributions of genetic and familial risk to premorbid profiles of adolescent eating disorders. And I have long contended, anybody that knows me knows that I have long been making the argument that the eating disorder field really needs to bring in rigorous and exceptional scientists from other disciplines, because the eating disorder field tends to train their own, and the discipline is largely focused or involves clinicians. And so we really need to build a stronger scientific base of neuroscientists and geneticists. And so when I crossed paths with Carolina, I was thrilled, because she was exactly the type of person that I had been looking for to build an eating disorders program, research program, both at UCSD and beyond. And so I think that you will hear from her talk that despite she's been spending the last year and a half working with eating disorders, her interests and her methods really extend to lots of areas of psychiatry. And so as she is starting to make her transition to the ROO phase of her kangaroo grant, we're really hoping that the DOP might be a good place for her to continue this work. So with that, you can take it away, Carolina.
Amazing. All right. Everyone can hear me okay? Awesome. Thank you so much for that introduction, Christina, and thank you so much for having me here today. I am really excited to share with you some of the bridges that I've been building over the last 12 years working in neuroimaging and in neuroscience, really harnessing multi-scale neuroscientific methods to understand the development of adolescent psychiatric disorders. So I am not from California or even the US. I actually grew up outside of Toronto, and I did all of my training in terms of my undergraduate degree and my PhD at McGill University in Montreal. But don't let the single institute fool you. I certainly was exposed to a myriad of different techniques and labs that really influenced my thinking. And for anyone that knows or has been to Montreal, you might know that it is really a hub for neuroscience. And in 2012, I just so happened to move to an apartment that was down the street from the Montreal Neurological Institute, or the Neuro. So I ended up volunteering there in a neuropsychological testing lab. And this is an institute that's so rich in history. This is where Brenda Milner, still to this day, walks the halls. She just did such in mapping the formation of memories through the hippocampus. Dr. Wilder Penfield carried out his famous surgeries here on patients with epilepsy to map the sensory and motor homunculus. So I really was exposed to such a rich, incredible neuroscience community. And around that time when I was volunteering, I heard through the grapevine that a lab that was led by Dr. Alan Evans, the McGill Center for Integrative Neuroscience, was looking for a research assistant to help out with a multi-site NIH-funded study called the Infant Brain Imaging Study, or IBIS for short. And IBIS is still going on to this day, and it's really trying to characterize very early risk factors for autism. So I was integrated into this neuroinformatics layer of the McGill Center for Integrative Neuroscience that really housed the data, curated all of the imaging, genetic, longitudinal data that was coming in from multiple sites. Now, this really was a center. So on top of the neuroinformatics layer, there was this image processing layer, and there was also this scientific layer where researchers in this group were really developing cutting-edge methodologies to try and understand neurodevelopment as well as neurodegenerative disorders. I was really interested in psychiatry, though. So just on the outskirts of town, there was the Douglas Psychiatric Hospital. And I did a short but very valuable stint of animal work, actually, with Dr. Cecilia Flores, where we carried out a mouse model of addiction. And we studied whether exposure to amphetamines during adolescence disrupted prefrontal cortical development differently than if you exposed a mouse during adulthood. And it turns out, indeed, it does. So timing really matters. But I knew that I really wanted to work with humans. So I went down the street still on that campus at the psychiatric hospital and found Martin Lepage's lab. And he was doing really great work, longitudinal imaging studies in parallel with clinical assessments in patients who had just experienced a first episode of psychosis. Now, Martin had just collected a 10 years worth of imaging data, longitudinal. And at that point, he was a little bit overwhelmed with exactly what direction we should take. So I saw a natural bridge to be built with the work that we had been doing with Alan Evans. And this became my PhD from 2014 to 2019, where I was really applying some of the novel analytic tools and neuroimaging methods that Alan Evans' group was developing and applying them to clinically oriented questions in psychosis. Now, somewhere in between, I had a couple conferences in San Diego, and it just so happened that Anders Dale in the Department of Radiology and also a member of the Department of Psychiatry, he had reached out to Alan and said, hey, we just started this massive study called the Adolescent Brain Cognitive Development Study. I know that you've been working on LORIS for a really long time. Is there any way that maybe you could just give us some advice or guidance? So no one had to tell me twice. I ended up coming to San Diego for a few months, teaching the group about LORIS. But in the end, I think it was more them teaching me about genetics, about ABCD, about neurodevelopment. And I absolutely fell in love with San Diego. So I finished up my PhD. It was a great motivation for that. And actually, to this day, the LORIS team is still consulting with ABCD, and it's also the backbone for the Healthy Brain and Child Development Study. So I feel really grateful that these collaborations are continuing. So in 2019, I joined UCSD as a postdoc, as Christina mentioned. And at that time, I was working with Chiwa Chen and Anders Dale and really trying to incorporate genomics into my neuroscience toolkit to understand the brain and in turn to understand complex human traits such as psychiatric disorders. And finally, bringing this full circle, last year, as Christina mentioned, I was awarded the K99 grant, and I'm trying now to really integrate all of this experience that I've gathered over the years in neuroimaging techniques and genomics and applying that to eating disorders. So I'll bring that back a little bit later. It might seem like I've jumped around quite a bit, and I have, but at the core of all of it, I've always really been interested in neurodevelopment and also really using tools in neuroscience that are kind of bridging across multiple scales to really understand complex human behavior. So this can go from the micro scale, such as genetics, single cell characterization, the mesoscale, such as cellular and myelin architecture, and then going towards things that we can non-invasively measure with magnetic resonance imaging, for instance, looking at regional structure and function, as well as how brain regions are kind of communicating or working with each other in a circuit level or systems level manner. And then finally, using all of those scales to understand behavior. So I do not say for a moment that I have done everything exhaustively, but I'm just going to show a couple of projects and examples of how we can bridge across these scales to get a little bit closer to understanding adolescent psychiatric disorders and in turn, perhaps informing treatment. Another really key theme that I'll be talking about today and that's been underlying my work is that timing really matters. So we know that adolescence is really a critical time window for the onset of many psychiatric disorders, as we see in this figure. And there seems to be something not only in terms of social and cognitive development, but in terms of brain development that is really important in these psychiatric disorders having their onset during this time window. So I really firmly believe that longitudinal measures are offering us something really valuable beyond just individual snapshots in time. So the first project I'll show you today is work that was part of my final chapter of my PhD thesis. And it'll be an example of how we can traverse the meso and macro scales to understand functional outcomes in psychosis in this project. So this was a paper that was published in Hippocampus in 2020, and we made the cover. So that's that cover pictured right there. So in case though, for those that aren't as familiar with psychosis, or just a very, very brief primer, typically what brings patients to the clinic are positive symptoms. So you can almost think of this as an excess of sensation or behavior, things like hallucinations, particularly auditory in nature, delusions. And antipsychotic medications are the first line of treatment for positive symptoms. They're actually quite effective in dampening down those positive symptoms. Not perfect, but quite good. But it's these other factors that I'm highlighting in red here, namely negative symptoms and cognitive deficits, that are really important for functional outcome and are really considered unmet therapeutic needs in psychosis. Strides are being made in the right direction, even since I finished my PhD, but there isn't a magic antipsychotic medication for things like amotivation, social withdrawal, and in particular, verbal memory deficits compared to all other cognitive domains tend to have the largest effect size differences between patients and controls. So we were really interested in understanding what was happening in the brain shortly after a first episode of psychosis that might be contributing to these negative symptoms and verbal memory deficits that we don't see in all patients, but in those that do have these cognitive and clinical profiles, like I had mentioned there, it's really important for functional outcome. So we worked with a first episode of psychosis early intervention or treatment program. So this was a clinic called PEP. You can see some of the cliniicians, and Martin Lepage was a neuropsychologist there. And what was really valuable is we worked really closely with the psychiatrists shortly after the patient's first episode of psychosis. So this is really acutely ill, to gather all of this clinical data. And alongside that, we had this neuroimaging protocol. And in this particular study, we actually obtained 4 neuroimaging time points within an 18-month time span shortly after that first episode of psychosis, because we were convinced that there were a lot of dynamic changes happening, not only clinically, but also neuroanatomically. And psychosis has been dubbed this disorder of disconnectivity, which can be really challenging when we're thinking about clinically actionable targets. So I turned to a really elegant theory by Carol Tominga that proposed that the hippocampus might be central to positive symptoms in psychosis. And she actually took, across multiple scales of neuroscience herself, she obtained evidence showcasing that there were lesions within this well-characterized circuitry of the hippocampus that might lead to hyperassociational memories and in turn positive symptoms. And we know that the hippocampus is a really key convergent zone. It takes in a lot of cortical input. Sometimes we forget that the hippocampus is cortical itself, it's ancient cortex. But it's also very heavily connected to the temporal cortex and prefrontal cortex, which are really important for verbal memory and some of these constructs of negative symptoms. So as I mentioned, we know that psychosis is a disorder of disconnectivity. It's not just one brain region that is impacted. So it's really important for us to take this circuit level approach. And finally, if we wanted to understand dynamic or plastic changes happening after a first episode of psychosis, I really turned to myelin as a marker of these sorts of changes. And I think sometimes we forget to appreciate that the highest number of myelinated fibers actually reside within the cortex. So this is what you're seeing in the schematic here in compartment A, whereas a lot of people usually study myelin within the context of long-range fibers. So we're still going to stick to the cortex here. So there were a lot of measures that we were interested in. So we had a really unique imaging protocol that I was really grateful that I was able to pilot this with Christine Tardif pictured here. So we had this quantitative T1 map that you see at the center. And this is a little bit different than your regular T1 weighted map, because this map actually, the voxels that you're seeing here, they hold a biophysical signal that is very closely related to myelin content. So you can compare across subjects and across time. And then for the hippocampus, we had this really beautiful submillimetric T2 weighted image where you could actually resolve hippocampal subfields. And we also used an atlas from a tool called Maggot Brain that Malar Chakravarti spearheaded. And you can find this openly available on GitHub. So you can see all these hippocampal subfields, but we also segmented some of the white matter sitting atop the hippocampus, such as the alveus, fimbria, fornix, and mammillary bodies. And then finally, on the cortical side, we just took a more conventional T1 weighted image and we segmented across 62 different brain regions, myelin content across different intercortical depths. We have this timing component. So I mentioned that there were 4 or up to 4 imaging time points per patient. So what we did was we turned to this method that we named dynamic anatomical covariance, or DAC for short. And this was really a method that was spearheaded in Alan Evans' group and also in collaboration with Buddha Kondrakpam and John Lewis, where essentially for each pair of regions and for across time points, we measured how similar or dissimilar are trajectories of myelin content between brain regions. And then we could do this for each of our brain regions of interest across time, and you ended up with this subject-specific matrix of co-occurring change in myelin that you're seeing on the right here. So the sample that we ended up in 7 patients with psychosis and 29 controls. I appreciate that this is a bit of a smaller sample, but just remember that this is a sample that has at least 2, if not 4 imaging time points. And we were able to, others in the lab were able to follow up with this work later on as well after I finished my PhD. So I'll comment on that again. And finally, we wanted to test this idea of hippocampal centrality. So we turned to graph theory and a measure called participation coefficient. And this can be envisioned quite simply. So imagine you've got a US network of airports. Chicago O'Hare is known to be very central to this network of airports, transporting people very efficiently across the country, whereas San Diego would have lower centrality compared to Chicago O'Hare, just because it has less of those connecting flights. Now, instead of airports, now we have brain regions, and instead of flight paths, we have these co-occurring change indices between myelin content between regions. And we can also start to group, for instance, cities together in a state. Very similarly, we can group brain regions together based on their function. So this is what we ended up doing. For instance, cortical regions underlying the visual system can be grouped together, or frontoparietal as well. Okay, so getting to the results, we did actually indeed find kind of bridging systems to behavior that we found lower hippocampal centrality in patients with a first episode of psychosis. And this lower hippocampal centrality was associated with both worsening negative symptoms over time and worsening verbal memory deficits. So these are those predictors of functional outcome in psychosis. And because we saw a lot of these pairwise relationships, we then put this together in a mediation model and saw that changes in verbal memory were actually helping to explain or mediate this relationship between hippocampal centrality and negative symptom change over time. And I've just been mentioning hippocampal centrality as kind of a whole, but when we actually looked post hoc, since we did have this really beautiful atlas, we saw that the results were really driven by myelin-rich output regions of the hippocampus, including CA1, alveus, fimbria, fornix, and mammillary bodies. So what we showed here, bridging the meso and macro scale, is that there were systems level alterations between neocortical and hippocampal myelin, and the hippocampus seemed to be a really central component of negative symptoms in relation to these myelin changes happening over time in other brain regions, and verbal memory helping to explain this relationship. So I worked quite a bit with psychiatrists during my time, during this project and also during my PhD. And if I started telling them about dynamic anatomical covariance and participation coefficient, their eyes would glaze over. And I understand these methods are really complex, but if we can give a more clinically actionable target. So if I say, well, the hippocampus might be really central to all of this, this gives us a more tangible target that we can use to understand some of the biological mechanisms that are underlying these important indicators of functional outcome in psychosis. So we were really excited by this hippocampal result because it is such a plastic structure in the brain. And this project actually served as the foundation for a grant that Martin Lepage was awarded a few years later. This is sort of our Canadian R01 grant equivalent from the Canadian Institutes of Health Research. And I'm really grateful to be listed as a co-investigator here as well. And we're actually now more comprehensively modeling these different neurocognitive factors in relation to functional outcome and also in relation to these dynamic brain changes across multiple sites in Canada. Okay, so we've talked about the meso and macro scale, and the next project I'm going to show is really integrating more of these measures at the micro scale. So this is including genomics as well as single cell characterization of regulatory sites. So I will break all of that down. And we actually know that a lot of psychiatric disorders and related traits have a genetic component. They're highly heritable. But I think it's a pretty big leap to move from genes to behavior, especially if we want to understand mechanisms or try and improve treatment. So my idea here, and this is not novel to me, but there's a lot of evidence suggesting that perhaps the brain might be this intermediate phenotype that we can measure between going from genes to behavior. And something that I've always found really fascinating and really striking is that if we compare the DNA of mice and humans, we actually share 92% of our DNA with mice. And we can obviously say that phenotypically we're very different. Our brains are very different. But it turns out that a lot of the ingredients underlying, for instance, cortical development are the same, but it's how these ingredients are regulated that are really changing and leading up to these really drastic phenotypic changes between humans and other species. So for instance, there's evidence to suggest that there's a much larger neural progenitor pool in human cortical development, and the cortex has had to adapt to really house these extra neural progenitors and has had to expand in terms of increased surface area, for instance, and this kind of buckling and folding of the cortex to adapt to that change. So we can actually measure things at the cortical level, such as cortical expansion, surface area, cortical thickness that you can see in the bottom left schematic here. We can measure this non-invasively with magnetic resonance imaging at a pretty cost-effective rate at this point in time. And what we did for this project that I'll show you that we published in Science a few years ago, and we also had some follow-up papers in PNAS, we conducted genome-wide association studies, or GWAS, where we're essentially taking many individuals. So you can see our data set to the right here, which I'll go over in a moment. But for those that aren't as familiar with GWAS, we have very similar genetic code along the genome, but every so often we get these single nucleotide polymorphisms, or SNPs. They're also referred to as common genetic variants, where individuals will differ by a base pair. So we're essentially taking that genotype at this common genetic variant location, or SNP location, and associating it with traits of interest such as cortical thickness and surface area. So now we're working with samples very different than what I was working with during my PhD. Over 32,000 individuals in our discovery sample from the UK Biobank. These are a group of older adults from the UK. We also had a replication set of over 7,000 individuals, really trying to integrate more diverse participants as well. And I've already alluded to the fact that I've been working with the ABCD consortium. So we ended up also including over 9,000 children from the baseline time point. So these are ages 9 and 10, to see if some of these findings from our GWAS of brain features were also generalizing to a younger sample, because typically GWAS have been conducted on these older adult samples, as it's just been easier to get the sample sizes we need. Another really key challenge here when we're thinking about GWAS of brain phenotypes are, how are we defining brain territories or brain atlases? And maybe you can find your favorite brain atlas there. But you can think of this as if you were, for instance, going on a hike. You would probably be relying on a map that looks at elevation rather than a map that is showing you the fastest highway to get somewhere, right? So if you are trying to understand the genetic variants that are associated with brain expansion or with cortical thickness, for us, it made sense to look at a genetically defined brain atlas. So that's exactly what we did. And we turned to these genetically informed brain atlases that Chi-Hua Chen had published 10 years prior to my postdoc that looked at these twin studies. This was from the VETSA sample. And mapped out these regions that, for instance, if you look at the blue region in surface area, this motor premotor region, all of the vertices or points or expansion of surface area in that blue region are going to be more genetically similar to each other. And the factors in the green regions, for instance, are going to be genetically distinct from the blue. So these are really these parsimonious solutions to understand genetic maps or genetically informed maps of surface area and cortical thickness. So these are the phenotypes that we'll be working with for our GWAS. I do notice something went in the chat really quickly. Oh, thank you. Okay, I will take a lot of questions afterwards for sure. So diving right into the results. So really bridging genes to brain structure. So we found, and what you see on these maps here, are all of the significant, so genome-wide significant loci associated with each brain region. I'm just putting the numbers here, but we'll put some interpretation on it in a moment. But what you can see right away is that we actually find more significant loci associated with surface area compared to cortical thickness. And actually, most of these variants that we found generalized also to our ABCD neurodevelopmental sample. So this was really nice to see. And also at the time, we found that the highest number of significant loci associated with brain structure compared to any other GWAS that had been published at the time. And I think this was in large part due to our genetically informed atlas. It's very hard to interpret the numbers on the left, right? So what we end up doing is we take those SNPs, we map them to genes, and then we take those genes and see what biological pathways they're a part of. And just focusing your attention for the bright red, to the bright red bars, sorry. So for instance, our visual cortex or occipital cortex had the highest number of hits for surface area. And then we can see functionally relevant neurodevelopmental processes that our SNPs were associated with. So for instance, things like pigment cell differentiation, melanocyte differentiation, and a lot of terms really important in early cortical development. A lot of the SNPs that we found were in non-coding regions of the genome. And as I had mentioned in sort of the introduction of this section, is we have a lot of the same regulation of these different genetic variants and how they are, how these genes are transcribed in different cells and at different points in development that are actually impacting these large phenotypic differences that we see in humans. So for this, we turn to collaborators in the Department of Bioengineering at UCSD with Kun Zhang and trainees who have moved on at the time, but super brilliant people to work with, Jenny Dong and Yan Wu. And we leveraged data that had already been published in human cortical tissue that mapped these accessible or open chromatin sites across 20 different cell types in the brain. And these open chromatin sites are essentially places where histones might fall off and transcription factors can actually bind, or there are these accessible sites for transcription factors to bind and then initiate downstream gene transcription. So for these cell types, they're all listed along the x-axis here in the left-hand figure, and then our brain regions on the y-axis. And you're seeing the significant enrichment for the SNPs that we found associated with our brain regions and these accessible chromatin sites in these 20 different cell types. Now, it's very hard to squint and see on the left, so the right schematic kind of shows you everything you need to know. So we found this significant enrichment for these open chromatin sites in microglia and layer 6 excitatory neurons with the SNPs that we uncovered in prefrontal regions, as well as links between parietal cortex and these regulatory sites and inhibitory neurons and astrocytes. And one of my favorites is this enrichment for these regulatory sites in oligodendrocyte precursor cells, which are precursors for myelin, that were actually associated with the genetic variants associated with a really myelin-rich cortical region, which is the motor premotor region. And one last finding here before I kind of summarize some of the methods we used here, really bridging genes to systems. It isn't that just one genetic variant is associated with a single brain region. We see a lot of pleiotropy, so there's a lot of genetic variants that are associated with multiple brain regions. And when we actually look at the correlations, both on a phenotypic scale and a genetic scale, we actually see that, for instance, with surface area, we see that the genetic variants associated with anterior regions of the brain are more similar and distinct from posterior regions of the brain. Whereas for thickness, we actually saw this dorsal ventral gradient of genetic architecture, where we see more similarity with dorsal regions in terms of thickness compared to ventral. And some of these SNPs were really interesting to dive into. We had about a quarter of our SNPs showing these sorts of pleiotropic effects. So obviously I can't go through all of them, but for example, there was one SNP that we found on chromosome 17 that was actually fine mapped to both a promoter and enhancer region for two different genes. And they actually helped to shape both increased area and increased thickness of orbitofrontal cortex, which I know is of interest to a lot of people working in psychiatry in terms of emotion regulation and a lot of the traits and brain imaging findings that we find with that region. So we found that this genetically informed atlas boosts discoverability of genetic variants underlying brain morphology. We're seeing that a lot of these, the cortical patterning is shaped by these regulatory genetic variants that are linked to specific cell types. So kind of bringing in this micro as well as meso scale. We're seeing also at the macro scale more of the systems phenotypes in terms of genetic programming, and we're seeing these genetic gradients early in development. And a lot of these regions that we're seeing in these genetic variants associated with cortical expansion are really important understanding regions that we have been discussing a lot in psychiatry underlying psychiatric traits. So there's a lot to sift through in, for instance, the science paper that I had just discussed, and just in any GWAS in general. It's so much data, and it can be really, I think, overwhelming to sit down and even read one of those papers, especially if you're not in the field. I'm also a first-generation academic, so I always have in mind really making science more accessible and making it also more clinically translational in terms of my experience working with clinicians. So we ended up writing an editorial that was published in the Journal of Psychiatry and Neuroscience. We wrote this with Chiwa Chen, but also Hao Wang, who I'm highlighting here because he is a pediatrician by training, and really gave some valuable insight into how we can best translate this to clinicians. And what we do in this paper is we highlight some of the genes that we found to be associated with our genetically informed brain regions and how those genes have been implicated in different neurodegenerative disorders and psychiatric disorders. So really trying to integrate this genetic, brain, and behavioral framework. And there's still, of course, a lot to be done in terms of clinical translation. We still need to boost discovery and replicability of these genetic variants. And also, we found so many genes, and a lot of GWAS will kind of showcase so many genes, and it's, I think, nearly impossible to go up and actually do really carefully controlled follow-up experiments. But I think there are certain genes that we can prioritize. So for instance, the DCC gene has been found to be associated with 8 different psychiatric disorders. This was actually a gene that was at the foundation of Cecilia Flores's lab when I worked there almost 10 years ago. So really trying to identify these genetic variants, especially those that might inform treatment response and how they're also impacting the brain. So for the rest of this seminar, I'll be talking about my kind of short-term and long-term research directions. And as Christina had alluded to, I'm really trying to bridge now some of these genetic findings and neuroimaging findings to the world of eating disorders. So this is a really sobering figure. It was published a few years ago, so that some of the landscape of this is shifting in a positive direction. But what we're seeing in this figure is that over 30 million individuals in the US are affected with eating disorders, more than these other disorders that are showcased on this figure, such as autism, schizophrenia, and Alzheimer's disease. But the amount of research funding that is allocated to eating disorders is so much smaller than these other disorders on the figure. So, and we also know that eating disorders, particularly anorexia nervosa, but also bulimia nervosa, have really severe health consequences. Anorexia has the highest mortality rate amongst all psychiatric disorders, only behind opioid use disorder. So this is really an urgent public health concern. And I am really motivated to leverage discoveries in neuroimaging and genomics to better understand the etiology of eating disorders. And there are some bridges that can be made with some of the work that that I've collaborated on over the years. So there was a recent, I guess not super recent, but 5 years ago, there was a GWAS that was published on anorexia nervosa. And some of the genes that were implicated in that GWAS, there was 3 genes that I'm listing in the middle of the right schematic here that we actually showed to be associated with expansion of different cortical regions, particularly parietal and temporal. And we also found these genes to be associated with restricted diffusion measures. So looking more at these microstructural features of brain connectivity and brain structure. So, and a lot of these genes also that were found in that anorexia nervosa GWAS are enriched for brain tissue. So this really motivated my K99R00 grant, which I started last year. And you can see my wonderful mentorship team on here, and you'll see some familiar faces from the department. So we're leveraging ABCD data as well as clinical data from the Eating Disorders Center to first just identify behavioral as well as MRI-derived morphological and microstructural features of eating disorders. Now, what's really cool about having both of these data sets is that they're pretty similar in age range, and the clinical data set that I am working with at the Eating Disorder Center actually has an ABCD imaging protocol. This wasn't planned, but I think this was the universe's way of saying this project needed to happen. And then finally, or I shouldn't say finally, but the next step in the R00 phase, I'm going to be incorporating more genetic tools, and hopefully also the Psychiatric Genomics Consortium might come out with more GWAS that we can leverage to really build this predictive multivariate model of polygenic risk, neuroimaging, and cognition and neurobehavior that might predict eating disorder onset in adolescence. So it's been a whirlwind year, and I'm just going to summarize some things that we found in the last year. But we have a preprint posted that characterizes disordered eating behaviors in ABCD. And what we're finding is that eating disorder symptoms are endorsed by both sexes. I think typically a lot of people think that they're female-only disorders, which is just not true. We're also seeing disordered eating being endorsed by youth that identify as sexual and gender minorities, as well as disproportionately high in youth who are identifying as Black, other racial minorities, Hispanic, as well as those coming from lower socioeconomic resources. So for those that work at the Eating Disorders Center, clinically, this is reflective of what people see in the clinic, but unfortunately in the research world, this sort of diversity is just not as present as it should be. So we're really excited that ABCD is helping us showcase the diversity of disordered eating. We're also seeing already at baseline, before youth are endorsing any sort of eating disorder-like symptoms, that they're particularly in the cognitive traits, there's specific patterns that are more predictive of, for instance, weight control versus weight distress versus binge eating symptoms 2 years later in ABCD. And at the level of the brain, we're seeing at baseline regions or morphometry of the corpus callosum, as well as caudate volumes being predictive of some of these later disordered eating behaviors. It's been also a really novel time for me to get into the eating disorders research field. And I feel like I've been so welcomed by the Eating Disorders Center. I can't see everyone on my screen right now, but I just really want to echo and say how grateful I've been to learn so much from the Eating Disorders Center. It's just such an incredible team and clinic, and they really have patient care at the forefront. And you can see an image of me at the top there presenting at my first eating disorders conference. I also have this unique challenge as part of my grant in bridging findings from a really large study like ABCD, and how are we going to generalize that, if at all, to a smaller data set? So I've been working quite a bit with Anders Dale and team to create these multivariate prediction methods using or leveraging ABCD study data and porting it over to smaller replication samples, as well as these considerations that we might have in leveraging these large consortia level efforts to power smaller samples in psychiatry. I've also mentioned that timing is a really big theme in terms of my research program. I've been talking a lot about adolescence, and I'm kind of bringing it a little bit earlier to this late childhood window with ABCD, but I've been really interested in understanding what's happening before this window, especially when we look at the genetic findings of brain structure. There's a lot of really early developmental processes that are happening that I think we need to understand in eating disorders. So I was awarded this Young Investigator grant from the Brain Behavior Research Foundation to bring this window of investigation even earlier in life. So we're leveraging over 100,000 trios from the Norwegian Mother, Father, and Child Cohort Study, or MOBA for short, where genetic data is actually collected on mothers, fathers, and the child. And the mother is followed from pregnancy all the way to postnatally, and then the youth and offspring are followed from very early in infancy all the way to adolescence. And already in this data set, we are seeing over 1,100 youth that have been diagnosed with an eating disorder. So we can look now at their genetics, at their familial history, at all sorts of variables spanning temperament, mental health, also metabolic factors, and how different profiles of these behavioral measures might be predicting an adolescent eating disorder, as well as whether genetic or familial risk might be differentially contributing to each of these profiles that we will identify. So for this, I'm working with Ule Andreasen, who's at the University of Oslo, and he's actually the psychiatric genetics lead of MOBA, and also continuing my work with Christina and Anders. As also really want to thank the trainees for helping me just navigate a Norwegian database. So you see Pravesh, Nora, and Victoria there. So moving this into sort of longer term plans, I really envision incorporating as much of these risk factors as possible to perhaps build this individualized risk calculator for eating disorders. And this is not a new concept. This sort of risk calculator exists in the psychosis world. And you can find that calculator at the bottom left there. That calculator integrates some clinical and cognitive assessments, but I can envision this bridging more of these multiple scales of neuroscience that I've been talking about, genetic factors, blood-based markers. I haven't talked about it a lot today, but I really want to incorporate more environmental factors, educational programs, community, peer support, et cetera, in trying to understand who will develop an eating disorder. And I think these risk factors are really important to understand the etiology of psychiatric disorders and perhaps early intervention, but I don't think it's enough. I think we need to find modifiable risk factors if we want to actually make strides in improving treatment and improving outcomes. So for instance, building certain learning programs that will be tailored to individuals' needs, building cognitive strategy programs to help with cognitive deficits that we're seeing, and in turn, how this might link to the brain and how some of these modifiable risk factors might be influenced by genetics. So sort of more of a slightly more concrete 5 to 10-year plan. I actually want to go back to kind of smaller, still well-powered, but smaller experimental designs where we can design these intervention studies to inform evidence-based treatment. So for instance, having sort of a pre and post MRI scan, and then in the middle of those scans, incorporating some sort of treatment or cognitive intervention, whether it's pharmacological, cognitive skills training, transcranial magnetic stimulation, and then measuring outcomes that we're interested in and that are important for patients' daily functioning, such as symptoms, cognitive flexibility, et cetera. And I think there has been some concern that neuroimaging is not necessarily predictive of behavior and that maybe we don't need neuroimaging to understand outcomes, but we are actually showing in some recent work that neuroimaging measures, particularly task functional MRI, can be predictive of task relevant outcome variables. So I think it's still a really important component of these study designs. And in turn, understanding how changes in the brain as well as changes in behavior, very similar to what I did during my PhD, might influence outcome. Now, you might have two individuals that undergo the exact same intervention, but have very different outcomes. So we will also need to take into consideration some of this complexity and how, for instance, genetic factors or medical history, peer support, community-based support might be also influencing outcomes in patients. So this is where I bring everyone in this department into this. There's so many of you already working at all of these scales. And I'm only incorporating some examples on this slide. Otherwise, it would be way too busy. But I know there's so many amazing groups that are doing work in genomics and psychiatric risk in the department. There's also people that are doing preclinical models of eating disorders and related traits. I also envision myself continuing to work with folks in the ABCD study that are measuring these longitudinal and multimodal measures of development, both cognitive and brain, and also finally bringing this into a clinically translational sphere. So continuing my work with the Eating Disorder Center, but I'm also really welcoming the opportunity to learn more about what's happening at the Interventional Psychiatry Clinic. And at the core of all of this is this incredible community within the department. So I already have become sort of an adopted member of the Early Career Council. So I've been on that council for a few months now, almost a year. Time is weird. So I have been able to learn about all of the incredible training opportunities offered through the PET Council. I'm also really interested in learning more about the DEI Action Council and just continuing to create these bridges, not just across neuroscientific fields, but between labs and also with our patients and our research participants. So kind of bringing this community a little bit more outwards. This is a picture that I took of a lesson plan that I led for patients at that take part in a psychoeducation group at the Eating Disorders Clinic. And here I was just teaching them about different brain regions and their role or potential role in disordered eating behaviors and thoughts. And patients had to kind of communicate with other brain regions through sticky notes. And I also really wanted to make sure that have been at the core of these psychoeducation groups is making science a bidirectional process. So just as an example, during that brain lesson, I also told patients about some of the typical stimuli that we see in a task functional MRI setting so that we can measure the brain or measure brain activity rather while people are viewing food stimuli that might be eliciting some disordered eating cognitions. And a very typical fMRI experiment that you see in the eating disorders research field is people just being shown some stimuli of food and being asked how anxious they feel. And I asked patients, well, how would you change this study? How would you make it more ecologically valid? And what they came up with was so valuable and also very feasible to actually implement in the scanner. So for instance, one patient said, well, rather than just seeing single food stimuli, we could be shown a video of someone preparing a meal, or we could be asked to imagine how we would feel after eating it, or stimulate more senses, especially smell. Maybe even cooking sounds would make this a lot more realistic. So once again, just really making this a bidirectional dialogue is going to make our science so much stronger. And bringing that community even further outwards, we know that science can only flourish with collaboration and diverse perspectives. And I've seen this firsthand through some of the work that I did for 3 years for the Organization for Human Brain Mapping, or OHBM. This is an international community of thousands of neuroimaging experts. And because there's so many of us, we kind of branch off into special interest groups. So I was part of the student and postdoc special interest group. And in 2021, when conferences were still virtual, I co-organized this symposium that we named All Hands on Deck, Patching the Leaky Academic Pipeline. And we included speakers that you see here, Lucina, Tenter Hinton Jr., Angeline Dukes. And the objective here was, one, to define the leaky academic pipeline and try and understand why we are losing disproportionately high rates folks from particularly underrepresented groups at sort of higher stages in the academic career pipeline. And Angeline was the first to say, I really don't like this metaphor. It's passive, but the system is actively pushing BIPOC scientists out. And this schematic on the right here illustrates this point, where you can see that there's this glass marble maze that's really engineered for the red dots to get through, and then the green dots get somewhere mid through, and then blue dots get stuck a little bit more behind. And just to put some numbers to this, and this is from a recent article that was published a few months ago, 81% of full professor positions in the United States are held by men, which leaves 19% of those positions for women. And in reality, there's actually less than 2% of those full professor positions being held by Black women. There wasn't even an exact number that was placed in the article. And this is really sobering, and we know that this needs to change. This is not representative of the actual demographics of Black women or underrepresented minorities in the US. So one tangible step that Attenter Hinton Jr. gave, which was really simple, but very, very effective, is just give people their flowers as their first step. Acknowledge people's accomplishments, give positive feedback, especially when you're an early career investigator or even a graduate student, undergraduate student. It's very easy to be tied up in these feelings of imposter syndrome. So something just as simple as, wow, I really like how you presented this during Journal Club can go such a long way. And then moving this also into setting up more safe and informal spaces for connection, just to get this conversation going, and just to learn from other people's perspectives. So when we could reconvene in person in Scotland for OHBM in 2022, I organized this lunch with mentors, and you can see the room is full. And at each of these tables, we have a mentor that's a leading neuroimaging expert, and all of these trainees around the table just eager to learn about that mentor's path. And we actually brought this to the ABCD consortium, where we actually host a virtual coffee, we call it a virtual coffee corner, where we essentially allow trainees to connect with other members in the ABCD consortium that they might not otherwise connect with, or might feel too intimidated to do so. And it's been really successful. We've been doing this for almost 2 years now. So this is my last slide before my thank you, and we can get to questions. I really envision my lab as building bridges between scientific fields, between scales, and also I don't expect everyone, I certainly am not an expert in all of the scales that I've talked about today. So really enabling this crosstalk and education between disciplines, encouraging creative scientific pursuits, and embracing diverse viewpoints, building bridges between community members across career stages, promoting and amplifying trainee voices and accomplishments, and integrating and elevating lived experiences. And finally, bridging both the science and the community to make science more accessible and inclusive. So I have a lot of flowers to give to a lot of people, people from Montreal and my collaborators here at UCSD in Norway. I've got collaborators in Australia and China that also helped out with the science paper, and all the trainees I've worked with over the years, and my own mentors and sponsors, funding. A huge thank you to all the participants, families, and patients who participate in our studies, and all of your lovely faces. Thank you so much. And I hope that I was not too long, but I'm really excited to take questions and continue the conversation. I'm going to stop my share so I can see people. Oh no, it was a little long. We started a bit late though.
Quick question, I'm sure you addressed this. Carolina, this was a fantastic presentation. I learned a lot. I enjoyed it. It was just phenomenal. Really quick question. So in your ROO phase, how much was genetics incorporated into the aims you're going to test there?
Yes, thank you. No, that's a really good question. I know there was so much content that I had to go through. So yes, I am going to be incorporating genetics into the ROO phase, and specifically it will be through incorporating polygenic risk scores, just because even though ABCD is a huge sample in terms of genome-wide association studies, it's small, which is so ridiculous to say, but it's true, right? So we will be incorporating polygenic risk scores from the anorexia nervosa GWAS, but also related traits such as OCD, neuroticism, and there also is a proxy GWAS of binge eating disorder, and maybe by the time the ROO phase, bulimia nervosa will have published their results. But yes, so we're integrating those, because then we can also port them over to the clinical data set that we've been working with. Christina Wirenga and Amanda Bischoff-Grieth are PIs on that project, and we have 150 patients that we can also calculate those polygenic scores.
Thanks so much. That's great.
Hi, I had a quick question. Again, amazing talk, Carolina. It's always great to see you present. I guess I may have missed this, but if you had mentioned this, but in your sort of like big picture calculator for eating disorders that you were talking about, where do you see comorbidity lying in there, right? Do you see it as a risk factor, or do you see also it as an outcome, given that condition like eating disorders is comorbid with a lot of other disorders? And I was just wondering what your thoughts were about that.
Yeah, this is a really, really great question. I think it applies to any psychiatric disorder, right? Comorbidity is the rule, not the exception. And I think that's mostly because of the diagnostic framework that we work in was not necessarily informed by biology, right? And there are ways, so it's a chicken and the egg problem. You don't know sometimes what comes first. For instance, with eating disorders, clinicians will tell you that almost every patient comes in with anxiety. So do you treat the anxiety first, or do you treat you need to do weight restoration first because that is critical. And then do you treat the anxiety afterwards? There are actually also methods using GWAS. It's called Mendelian randomization. It's not perfect, but you can actually use these common genetic variants to try and understand causality between different traits. I don't know if one has ever been done for like anxiety and eating disorders, but for instance, like with metabolic traits, there's some evidence to suggest that the metabolic factors are coming before the eating disorder. So yeah, I think I think some of those risk calculators will capture those risk factors. And then I think it'll start to bleed into other diagnostic categories naturally. So does this become just like one big risk factor for a P factor? I don't know. But I think we need to start somewhere. And already eating disorders is transdiagnostic in nature, but they're more genetically similar than some other conditions. So I think time will tell, and I think more GWAS will also be able to help us elucidate what comes first. But yeah, this is such a big question in psychiatry. I appreciate it, Harry, and thank you for coming.
Of course, no worries. And thank you for answering my question.
I have a question. Great talk. I don't know if you know me, but I run the early psychosis program at UCSD and was actually one of the authors on the psychosis risk calculator. So anyway, you've kind of merged some of what I'm really interested in. Actually, eating disorders was my first love, but when I first came to UCSD, we didn't have an eating disorders program, so I ended up in early psychosis. But I know that as we've had these models of early psychosis, I've always kind of wondered, whether there could be a comparable model in eating disorders. And I remember I was on a study section with, I think, Eric Stites years ago. And I said to him, I said, well, what are the early signs? Because in the prodromal psychosis area, we identify people before the onset of the disorder. And so the psychosis risk calculator was developed with people who already had like attenuated symptoms, or they had a family history of psychosis. So we already knew they were high risk. And so we would say, okay, well, who among this group has the absolute highest risk? And so I was, so I had said to him, what do you see? And, his only thought, he goes, well, maybe picky eaters, or, and I'm sure that Christine and others have ideas about what some of these early symptoms look like. But, because I know that it's highly inheritable and you see a lot of, but have you thought about like how you would want to apply that? Because I know that within the psychosis field, you can't really apply the calculator to the general population. There's too many false positives if you try to do it there. So you already kind of need the high risk, or you might be looking for some outcome that is, a worse outcome among the eating disorder population. But have you thought about how to model that?
Yeah, thank you so much for that question, Kristen. And I did not know that you would be on the call, but I'm glad that we made that connection.
I'm glad I was able to join today. It was great, wonderful talk.
Thank you. Yeah, no, this is such a great question. I always sometimes worry when we're talking about these like premorbid risk profiles. We don't want to over pathologize everyone. There's also a risk just in telling someone they have risk for something, right? So no, I don't think that we're going to be screening every single individual, but are there, measures that we can take from health records? So for instance, I'm going to go back to the metabolic factors, because more and more we're seeing that these are really genetically correlated with eating disorders, and that they might come before. And in a lot of eating disorder cases, you'll hear cases that, okay, they already had this like fast metabolism growing up and they didn't eat as much food, and maybe it's picky eating, but it might be also other things on top of anxiety, on top of maybe some familial history of either an eating disorder or OCD or anxiety, etc. So if we could start to pick up, maybe that'll be our clinically high risk from, I don't know if we're going to be able to get, medical records like that easily, but already kind of flag someone based on, I mean, that's, I guess, the point of the risk calculator, right? Is we can flag just based on variables that are entered in. Okay, they already have low BMI, they have this sort of, HDL or lipoprotein profile, and they're really anxious, perfectionistic, and they meet some of these levels, and they also have a mom with anorexia. Okay, maybe that should be flagged and followed up. But definitely, I always am cautious about over pathologizing. Right? There's always that risk. But I think the early psychosis or the psychosis world has been a really great model in terms of successful early intervention programs. So I actually find, even though I did not plan it this way, I often find myself going back to some of the literature that I worked in with the PEP clinic in Montreal. Even the antipsychotic medications that were prescribed there are being prescribed to patients with anorexia now. So I think is a lot that we can learn from the early psychosis field. So I'm really happy you're here. And I think we also need people like that are transdiagnostic and bringing that expertise to other much needed fields as well.
Right. And I think one of the challenges, another challenge you brought up a lot of wonderful parallels to use when, but one of the challenges with a lot of the biomarkers, trying to get them so that they're user friendly and translational into the clinic, because how many general hospitals can do an analysis of brain connectivity to be able to use that in a calculator to help recommend treatment. So it's like figuring out how can you get these tools so that they can be done in the clinic. So a lot of the reason we ended up coming up with, say, neurocognitive biomarkers is that those are pretty easy to administer in the clinic. Because some of like the imaging biomarkers, you need serial scans in order to look at whose gray matter is thinning more quickly or something like that to put into the model. But those are the interesting challenges of the field. But anyway, I'd love to, if you ever want to talk about some of these ideas that you're developing, because I love it that you're going in this direction.
Appreciate that. I'd love to chat further.
Sure.
I do realize it's past 5 already, but thank you.
Okay, well, since it's past 5, thank you all for attending this talk, and I'm sure Carolina would be more than happy to field any questions offline if there are questions that occur to you later on. But yeah, I think I just share everybody's enthusiasm. I'm looking at all the comments in the chat, Carolina, and they're all fantastic. We all feel that this was an excellent presentation, really, really significant, impactful work, and just so excited that you're applying these skills to psychiatry and eating disorders.
Thank you. Thank you, everyone, for being here, and hopefully we'll cross paths in person soon. Bye all.